Professor Schumann.
Tell us a bit about the research interests in your laboratory.
So we're using imaging technology to assess mostly about glaucoma, and we're covering a broad spectrum from identifying the disease with new technology, new way of analyzing the data, better understand the pathophysiology of the disease, and all the way to track the disease changing, what is the best way of identifying changes over time. We are using different imaging technologies, and we are involved in developing new technologies. So it's a broad spectrum of stuff that we're doing. The silver lining that connecting everything is the goal of improving detection of glaucoma and assessing how the disease is changing with the better understanding what is happening, all through using the imaging technologies.
Broadly speaking, what are the biggest clinical challenges in glaucoma? Is it early diagnosis? What would you say it would be?
So glaucoma is mostly asymptomatic until relatively late in the disease. So early detection of the disease is a major challenge. A patient don't experience anything until they are practically losing most of their sight. The damage is irreversible. So the challenge is to identify the damage as soon as possible and as accurate as possible. So this is the first challenge. We know that for every patient that we identify of having the disease, there's another patient walking around undetected. So this is a major implication of proper identification of the disease in an early stage of the disease, especially considering there is a good treatment that mostly halt the progression of the disease. So as soon as you identify the disease, start treatment. If you have a successful treatment, you can prevent further damage and you preserve vision for long term. So this is a major task. The other aspect, which is very, very challenging, is to identify, once the patient already have the label of having glaucoma, identify changes of the disease. The disease is typically slowly progressing disease. Clinically, to diagnose those changes, it's challenging. We're talking about really small changes. And it is very, very difficult to do this. Having the ability to detect those changes accurately, quantitatively, it's a major advantage of using imaging technology. So this is one of the other major challenge in clinical practice of managing glaucoma patient, that the use of imaging technology can really advance our clinical ability.
So tell us how your work is changing those parameters of early detection.
So we have actually, our contribution is in different levels. So we are involved in developing new technology that are more sensitive to early diagnosis or early detection of disease. We're involved in developing a new method of using the information is acquired with those imaging technology to better identify those early changes. Image processing, a sophisticated way of processing the raw signal coming from those technologies. And for each one of them, we have our own publication that report of this work. We have multiple studies reported and still in the pipeline, looking at ways of diagnosing or identifying changes in the disease over time. So, for example, we have a cohort that span up to 18 years of follow-up with both structural information, so images that were acquired with optical coherence tomography, OCT, over a period of time of up to 18 years. So this is the longest available quantitative information that exists using OCT on glaucoma patient. We have all the functional testing, all the clinical information about those patients, giving us an unique opportunity to really identify what's happening over a very long period of time with the way of quantifying those minute changes. And that's very unique, and we are actively working on this data. We are working on a project that tries to understand what is happening at the level of the lamina cribrosa with animal models, to better understand what is the reason why glaucoma starting in certain eyes, where other eyes which have somewhat similar behavior properties don't develop glaucoma. So better understanding the interaction between forces that coming from the intracranial pressure and forces that coming from the intraocular pressure, both sides of the lamina cribrosa within the optic nerve head, to better understand what is happening in glaucoma. So here I'm covering multiple things and multiple aspects that we are doing in our group, trying to address those different aspects that are critical in my mind to understand and improve, at the end of the day, improve the understanding and caring for patients.
So you're obviously using new techniques or a new iteration of OCT, is that correct?
Yes.
And what specific enhancements have you developed to help you in this process?
So we can divide it into hardware and software development. So hardware, it is not our own contribution. We are not engineering this aspect, but we're collaborating with different groups in developing new technologies. So, for example, a new generation of OCT with new capabilities of OCT. So faster OCT, better penetration into the tissue, allowing us to do through certain manipulation a more reliable scan, that giving us a full visualization of the area of interest, with less effect of the deterioration of the signal through the thickness of the tissue. So this is different hardware development, and again we are working with groups that develop this, and we are implementing it in the clinical setting and use it to improve our diagnostic ability, as I mentioned beforehand. The other aspect is the software development. So there are people in the group that are expert in image processing, manipulating the image for better identify property in the signal that would be relevant for early diagnosis or early marker of disease, identify changes that appear in the signal, better analyzing. So we have also a statistician in our group that look on ways to use the information, but through innovative, advanced statistical method, come up with detecting what is real changes that are occurring over time. So we have different aspects to try and answer those questions.
And where are you in the process of putting this all together into a new and improved approach that other clinicians can follow?
So the things that I just mentioned to you, some of them is already in writing. So some of the information is already published in peer-reviewed journals. Some of the information I told you, some of the projects I told you that are still in the pipeline. We're working on them on different stages. Some of them are in the early phase. Some of them are at the writing process. We try to make the information we generate, or the innovative stuff that we are finding in our research, to make it as translational as possible. The ultimate goal of our research is to be able to translate to the patient, the glaucoma patient, or patient that is suspected of having glaucoma, so it will be impactful on managing the glaucoma patient. And this is why, actually, this is our primary goal. So we're doing it because we are, as scientists, we are curious to know what is happening. But at the end of the day, we want it to be translatable to managing a glaucoma patient. So this is either by reporting it as soon as we can, coming to conferences like that and try to disseminate this information. We're going to other meeting, and so on. So this is the way we try to disseminate the information.
So ultimately, you see the development of a new gold standard protocol that other clinicians can rely upon to get the very best diagnosis and follow-up using OCT.
Yes, yet this can be challenging. And I think glaucoma is a very good example for this. For example, what to define as disease progression when you are using advanced imaging technology like OCT to define progression? We have very hard time to come up with a definition of progression and would be widely acceptable as the gold standard. This is structural progression. Is it 2 micron? Is it 3 micron? Is it 3 micron over a period of time of 3 years, of 2 years, of whatever it is? How many times I need to repeat the test to make sure that the changes I see are reliable? We don't have clear-cut criteria. On the other hand, there are fields that this really make a difference. We can come up now with a clear-cut criteria. This what we call change. This is what we call disease. Here, the imaging technology with giving us reliable, accurate, reproducible information, it really make a change for certain region, for certain aspects, of clinical care.
What are your next steps currently for the work that you're progressing on?
So it's basically keep moving on the different projects that I mentioned to you beforehand to further understand what is happening. So if I mentioned to you the understanding the interaction between the intraocular pressure, the intracranial pressure, better understand what's happening in the short term, what's happening in the long term. So right now we are more focusing in the acute aspect. Glaucoma is a chronic progressing disease. It takes time for the changes that are occurring in the eye to fully develop. But this is a matter of time. We need to develop our level of understanding in a sound footing, and that's what we're doing. So we're starting with the acute, and gradually we're building from that as kind of a body of evidence to move forward. So I just give you one example what I mean by moving forward on stuff that I mentioned beforehand. So this is one aspect. Another aspect I mentioned to you beforehand is using the long-term data that we have. This is a huge amount of information that we really need to dig in to better understand what's happening in the disease. So we are investing a lot of effort into it. We're investing effort in trying to come up with a way to identify a patient that would be at high risk of developing disease, high risk of developing aggressive disease. So we are calling it stress tests. So doing something similar to what we are doing in cardiology, so you're exposing a patient, or you're putting a patient to run on a treadmill, doing all different exercise to see how the heart respond to this stress test, and then from the response you can tell, this person is at high risk and whatever. We're trying to do something very similar, a simple test that would tell us these are patient subject that would be exposed, or most likely would be the one that would go in the route that would lead to aggressive disease, and these are the subject that most likely have the property of the eye, whatever that is, have the property of the eye that would keep them healthy with full vision through their lifetime. So these are just some example of the things that we're currently working. I do see them coming up in some of the near future, some of them more on the long term. And again, I strongly believe that this would have a strong impact in management of glaucoma patients and people that are suspected of having glaucoma.
Glaucoma in a sense is like hypertension. It's a silent killer. Hypertension is a silent killer, and glaucoma is a silent stealer of sight. So ultimately, do you see the OCT moving out beyond ophthalmologists into screening centers, into primary care practices? If we are to make an impact, is that a direction that we should be thinking about?
Potentially. And you mentioned one example. I can add other example, for example, Alzheimer's. So completely different aspect, neurology. The eye is considered an outpouch of the central nervous system. Here we have a potential to go and see what is happening in the central nervous system, non-invasively, reliably, repetitively. Those imaging technologies don't cause any problem or damage to the eyes, so we can keep scanning it. Potentially it have a huge impact in medicine, in certain level of or certain aspect of medicine. Hypertension, there are studies that have shown the ability to measure the blood vessel in the eye, the ability to show the changes in the blood vessel caliber in the different stages of hypertension. Yeah, it has an impact. It's a matter of time until it would be incorporated more in other discipline of medicine. There's still way to go. We still need to make it a very robust technology, cheap, easy to operate, but we're making a lot of progress in this. The price of the technology nowadays is much less than what it used to be. We have many more devices that are available out there. There are many more companies that are interested in this technology, which mean that I would expect to see in the future, not even in long-term future, I would expect to see it incorporated in different aspects of medicine and help us to manage the patient not only for ophthalmology but with a broader application.
So you'll see that there will be a minimum set of capabilities for the OCT units, that they will be able to fulfill all of the requirements that you're seeking for. So do you see the development of a spec sheet, in a sense, that manufacturers would have to use as a minimum data set to be able to meet the standards that you're proposing?
Yes, and you do see it already today. So there are different devices. Each one of them is, not each one of them, but you see a couple of them are designed with focus of certain niche or answering a certain niche. I'll give you one example. So there are a couple of devices available with the which are specifically designed for research. So they have full flexibility. You can do whatever you want. There are handheld devices that are exactly what you mentioned. So they are limited capability. They can do only a certain thing. You don't have that robust, large normative database as included in the full-fledged, full capability device. But this would be a device that it is easily to operate in an operating room setting that would give you only this kind of measurement that might be relevant for the operating room setting. On the other hand, the other device that would be, I don't know, in the primary care office, this one includes everything, so it would be for glaucoma, it would be for blood pressure, it would be for Alzheimer's, it would be for diabetes, it would be for whatever you can think of. Yes, and you already see it tailor in, for different use right now.
You mentioned application for Alzheimer's. Are you actually able to visualize the beta amyloid using OCT?
So it's not really visualizing them as such, but there are indirect indicator that are associated with the neural damage that you can see in the eye that has been shown in some study to be related to the disease and the severity of the disease. I have to be cautious with this because there are some also conflicting information. Obviously, this can be a difficult population sometimes to work with. The information is sometimes the quality is sometimes so. Again, there are a couple of studies that have shown some damage to the neural tissue that you can detect with OCT that has been shown to be associated with the disease.
Is there anything else that you'd like to share with us about your program and your objectives going forward into the future?
So I'm a strong supporter of the incorporation of imaging technology, whatever it is, and I don't have any invested interest in any of the technology, but the imaging technology, the ability to provide us quantitative information, high quality visual information, significantly change the way that medicine is managed in ophthalmology. And I do see it in the future becoming even more integrated in the management of patient, not only in glaucoma, in other aspects of ophthalmology. And someone mentioned to me, and I like this similarity, that he see the incorporation of imaging technology in ophthalmology, the impact of it is similar to what happened when CAT scan was introduced to general medicine. We are in a phase where the imaging technology really revolutionized ophthalmology. And this is actively happening right now, and I still see it moving forward to further improving our ability to manage our patient. And I strongly believe that this would make a huge impact, not only what we accomplished so far, but this would be just the introduction of what's going to happen to us in the near future.
Is OCT useful for looking at the viability of stem cells or gene therapy?
Interesting question. We start to look into it. The question is complicated because the stem cell project right now depends on how you're doing it. So if you are doing it by injecting stem cell into the area of interest, exactly like what you're showing to me, injecting into the retina, the ability to quantify the retina with the imaging technology, this with high precision, and we can do it repetitively. This is a very strong tool for us to really see at least how they incorporate into the tissue. So this is one way of doing it, but the other potential way of doing it is to conjugate those cells with a signal enhancer. So conjugate them with a material, with contrast agent, to improve the signal or allow us to detect them and where they go, with the technology that, again, it's non-invasive, and we can do it time after time to track those cells where they go. Sometimes it sounds like science fiction, but we are making progress in this direction. So I'm not sure if we can do it at this point on a single cell level, but definitely we are moving toward the ability to detect those changes and how the stem cell is really getting into the tissue. And these are stuff that is happening right now.
Professor Wollstein, thank you for your innovative and groundbreaking work, which will have impact not only in ophthalmology, but in all fields of medicine.
Thank you. Pleasure to be here.